Hello and welcome back. Today is all about the ultrastructure of cells, and what that means is what's inside a cell? What does it do? Why do we care about it? And why do you need to memorize it? So let's get started. How do we even know about all of these things? Well, of course, microscopes, right? That's what microscopes do. They make microorganisms be visible to us. I'm sure other things could look at it too, but we're the only ones that write it down. So there are types of microscopes. The light microscope is typically what you see in a classroom. So all of the microscopes that you've really been exposed to in your science career is a light microscope. That is limited. So it's limited by the wavelength of light, which is why we call it a light microscope. So the maximum resolution of that is only 0.2 micrometers. And that's not, that's good, but it's not as good as it could be. And that's where electron microscopes come in. So these allow us to see the ultrastructure of cells. So look at the difference here between like a light microscope. Yeah, you can see stuff. Like clearly I can see the cilia that are being, that are protruding out of the cell. But then look at this. This is so clear. This is such a better image. And that's because of electron microscopes. Okay, so the resolution is 0.001 micrometers or a nanometer, which is much more intense. It's much closer. You can really see the fine lines between whatever it is that you may be looking at. The downside to an electron microscope, though, is that the specimens must be in a vacuum, so you cannot view a living organism under an electron microscope. You can view a living organism under a light microscope, though. And then there are two types of electron microscopes. This is like a subsection of electron microscopes. There's transmission, and then there's scanning. So transmission sends a beam of electrons through the specimen, and then scanning scans the surface of the specimen and shows you what ever is on the surface. Okay, so transmission is for like the insides, and scanning is for the outsides. Now, let's talk about the cells themselves. We're going to start with the older, less complicated cell, the prokaryotic cell. So this is an image. You should draw this. So within a prokaryotic cell, you have pili, which are these protrusions. You have ribosomes, which are these dots, the capsule, which is like the green outer layer, a plasma membrane, which is like your cell membrane, flagellum for movement, the nucleoid region, which is your DNA, cytosol, which is like cytoplasm, and then the cell wall, which is this like outer membrane around the plasma membrane. Okay, so once you have that drawn, we can talk about what all of these things are, right? So the capsule, the slime capsule, is for protection, and it's for protection against drying out and from being eaten because bacteria are small and can be eaten from like accidentally. The cell wall is a rigid outer layer that maintains its shape. It's again for protection. The cell membrane controls entry and exit of substances. Cytoplasm contains enzymes for metabolic reactions. Nucleoid is the DNA area, and then the plasmid itself is additional DNA molecule that can exist and replicate independently of the nucleoid DNA. What's awesome about the plasmid is this can actually be transmitted between bacterial species, and this is how some bacteria can learn new, like create new proteins that they couldn't previously make because they have accepted this plasmid from a different bacteria, so it can kind of do new things without evolving over time. They sort of like, it's like a cheat sheet to like learn new things. Okay, these are important structures that you should know and be able to annotate on a diagram. So please make sure that you know all of these structures. Continuing on with the prokaryotic structures, we have the ribosomes, which make proteins. In prokaryotes, ribosomes are smaller than ribosomes found in eukaryotes. So in prokaryotic cells, they are 70S, and eukaryotic, they are 80S. The flagella is for movement, and then the pili are those hair-like extensions which can serve one of two roles. One is attachment, which allows bacteria to attach to one another, and that's how plasmids can transfer from one bacteria to another. Or they can use it as reproduction, and it's called the sex pili, and it's longer in length, and this is for bacterial conjugation. Bacterial conjugation is the exchange of genetic material, so this is a way for bacteria to go through sexual reproduction, which is not usually what you think of when you think of bacteria. Normally there are just like replicating clones of each other, but they can actually go through sexual replication, and it is called conjugation. The asexual form of bacterial replication, though, and reproduction, is called binary fission. And this is when DNA is just copied, and then they adhere the DNA to different parts of the cell, and then the plasma membrane starts to divvy up the cytoplasm, and the cell elongates and then divides in half, splitting both the cytoplasm and the DNA into two identical cells. Now, we talked about prokaryotic structures. Most of it might be slightly familiar to you, but there were definitely some new structures within there that you've not heard of before. Now we're going to transition into eukaryotic structures. So this is basically like all the organelles that you have maybe heard of at some point in your biology career. So diving right into this, we have the nucleus, which is where DNA is found. It contains genetic material. All of this, I know there's like so much information. All of this could potentially be on an IB exam. It could be in class, so I do suggest actually writing all of this down. The nucleus has a double membrane, and it has pores which allows the mRNA to travel in and out of the nucleus. The nucleolus is a small organelle that is inside of the nucleus, and it produces ribosomes. Ribosomes are responsible for protein synthesis, and we'll talk way more about that in the next unit. But ribosomes are made from two rRNA subunits. The R in rRNA stands for ribosomal RNA. And ribosomes are structurally different in prokaryotic and eukaryotic cells. We talked about this on the last slide. Prokaryotes have 70S ribosomes, eukaryotes have 80S. You don't really need to know what the S stands for, but it is in reference to how quickly molecules move in a centrifuge. So prokaryotes move quicker than eukaryotes. Additional information about ribosomes. Free ribosomes make proteins to be used in the cytoplasm, and bound ribosomes on the endoplasmic reticulum make proteins destined for specific organelles or outside of the cell. And then the rough and smooth endoplasmic reticulum are for transporting materials. So rough and endoplasmic reticulum makes proteins. Smooth endoplasmic reticulum deals with lipids. It can also detoxify drugs in the liver or store calcium. Both vitally important. There's the Golgi apparatus, which is responsible for receiving, modifying, packaging, and shipping different products. So these might be things that were from the endoplasmic reticulums, and now the Golgi is responsible for actually moving those vesicles wherever they need to go. So there are two sides to the Golgi apparatus, the cis side and the trans side. The cis side receives whatever vesicle it is and receives products from the endoplasmic reticulum. The trans side is the shipping side, so it's the opposite side of the Golgi, and this is where products are actually discharged in the vesicles. There's the lysosome, which is just like where waste products go, right? It's a sack of digestive enzymes. It digests all the major groups of macromolecules. That includes carbs, lipids, proteins, and nucleic acids. It basically is just there to break down waste products and remove waste products. Vacuoles and vesicles, they store food, waste, toxins, and water. There's large vesicles that are formed from the Golgi. There's also a difference in size in vesicles and vacuoles in plant cells versus animal cells. Mostly it's there as like a storage closet, so this is where anything that the cell might need or is holding onto until it can be released, this is where it will be held. And then for energy purposes, there's the mitochondria and the chloroplast. The mitochondria is in all cells, generates ATP from cellular respiration. It's similar in size to bacteria, contains its own DNA, can self-replicate, and has inner membranes called cristae. We will talk much more about the mitochondria and the specifics, but you should be aware that this is where cellular respiration happens, and it's the formation of ATP. Chloroplast is in plant cells and is the site of photosynthesis, so this is how plants get energy. It only occurs in algae and plants, has its own DNA, similar to mitochondria, and 70S ribosomes, similar to what we see in prokaryotic cells. And its inner membranes are called thylakoids. And then finally, our last few bits of eukaryotic structures, we have cilia and flagella, which are used for movement, the cell membrane, which controls what comes in and out of the cell, and then the cell wall, which is for support and structure. So these are really important, like compare and contrast. We're going to talk about prokaryotic versus eukaryotic, and we're going to talk about plant versus animal on the next slide. So pro versus eu, in prokaryotic cells, the DNA is in ring form without protein, but in eukaryotic cells, it has proteins. In prokaryotic cells, DNA is free, but in eukaryotic cells, it's enclosed within the nucleus. Prokaryotic cells have no mitochondria and 70S ribosomes, and they do not have organelles. There's no internal compartmentalization to form organelles. In eukaryotic cells, they do have mitochondria, they have 80S ribosomes, and they have many organelles within. And then, of course, prokaryotic cells are smaller than eukaryotic cells. When we're talking about plant versus animal cells, in plant cells, you have a cell wall, and in animal cells, you do not. In plant cells, you have chloroplast and a large vacuole for water storage. In animal cells, there's no chloroplast, and the vacuoles are present, but small. Sometimes there's not even vacuoles present, but if they are, they're much smaller in relation to plant cells. In plant cells, you have carbs stored as starch, and they do not contain centrioles within a centrosome area, where in animal cells, the carbohydrates are stored as glycogen, and they do contain centrioles. And finally, plant cells are rigid because they have that cell wall, so it has a fixed, often angular, usually like a rectangle shape, whereas animal cells do not have the cell wall, so they are much more flexible and more likely to be rounded. Last but not least, we have the outermost part of all of these cells. So in bacteria, they do have a cell wall. It is made up of peptidoglycan. In fungi, they also have a cell wall. It's made of chitin. Chitin is the same thing that is made up of an insect's exoskeleton. You know when you like step on a bug and it makes that crunchy sound? That's chitin, and that's what fungi's cell wall is made up of. Yeast have a cell wall of glucan and mannan. Algae have a cell wall of cellulose, which is the same thing that's found in plant cell wall. And then animals, of course, do not have a cell wall. We just have the plasma membrane as our outermost part. And it does secrete a mixture of sugar and proteins, though, and that's called glycoproteins. And it forms an extracellular matrix around the cell. So that's sort of a little bit of extra protection that animals have since they do not have a cell wall. All right, great job. I know that was a ton of information and a ton to write down, but Promise you it's important, so I'm proud of you. I'll see you in class.